So, in this module I will discuss that the case study of another biomedical prototype development, and that is that zirconia toughened alumina based femoral ball head. So, these zirconia toughened alumina based femoral ball heads, as I told you in the last module, that this particular femoral ball head can be made up of either cobalt chrome or stainless steel or ceramic, and then acetabular liner, acetabular socket, this is typically made up of ultramounted polyethylene. So, this one I am saying that we are planning to manufacture this femoral ball head with zirconia toughened alumina, which is abbreviated as ZTA, and ZTA is popularly used in the ceramics community as well. So, what you see here is that is a typical total hip joint replacement here with this neck of the femur as well as femoral ball head, which is fitted there, and with an acetabular socket. And I mentioned in the last module also that femoral ball head typically made of cobalt chrome and acetabular socket highly cross-linked ultramounted polyethylene. Now, this kind of combination, they do not have the best possible wear resistance property. So, therefore, the research worldwide is looking at alternative to the currently clinically used femoral ball head material, and one of the potential material or potential class of material is bioceramics. Bioceramics means those ceramic materials which can be used for biological or biomedical applications. Now, one of the classic ceramic material that people use not only for biomedical applications, but also for a large array of fundamental study is that alumina. So, alumina is a very classic ceramic material, and it is well tested for different type of properties and so on. So, having said that, we have one would kind of focus or one would try to see that how alumina can be adapted in this kind of environment. But the problem with alumina is that despite various other properties which are typical of any ceramic material, alumina is not the best of the toughest ceramic material, because the toughness is a major issue of the ceramic, and then alumina has a very poor fracture toughness like 3. Whereas zirconia is one of the candidate ceramic material with relatively large toughness. Large means among the ceramic materials, but large certainly not as compared to metallic materials, because zirconia can have fracture toughness of 10 MPa square root meter, which is kind of relatively a respectable number in the ceramics community. Now, in order to mix that, or in order to kind of toughen that alumina with little bit of zirconia, you can produce the zirconia toughened alumina material, and in the total heap replacement, when these particles are released, as can be seen here. Now, these are the major culprits, because these particles can do all the different biological, or these can lot of significant or biologically significant actions in terms of macrophages and giant cell activations, and that will lead to inflammatory mediators release and osteoclasts activation. Osteoclast activation means at any point of time in any bone, healthy bone, if you have more osteoclasts than less osteoblast, then bone resorption will take place, and ultimately that leads to aseptic loosening. So, this is the major failure mechanisms currently understood for the total hip joint replacements, and as a result the revision surgery is the only alternative as far as that clinical option is considered. Just to give you that how good or how badly that clinical fraternity they need this femoral ball head development. In India itself, according to the total arthroplasty joint statistics, that 6 lakhs of total knee and 3 lakhs of the total hip replacement is there, is currently conducted. And we have also discussed in the one of the earlier modules that we have developed this some of the new material for acetabular socket HDP HAP alumina. So, this is essentially existing ultramounted polyethylene socket, and so what we have done? We have done essentially the lab scale research just to produce different simple shaped materials. So, this is your kind of 10 millimeter diameter cylindrical shaped, then you can do this 20 or 30 millimeter cylindrical shaped sample, as well as some rectangular bar of 40 to 50 millimeter. Now, all these materials of simple shaped were developed to measure a number of material properties like hardness, fracture toughness, wear resistance, and or initial cell compatibility study and so on. But the real challenge is to make this kind of geometrically complicated shapes, which is not very easy, and often you cannot use the lab scale processing conditions or lab scale sintering conditions. You cannot adapt the same lab scale sintering conditions to fabricate this kind of acetabular socket or femoral head. So, one has to tune or tailor the lab scale conditions to produce this kind of geometric shape. Now, this fabrication of the ZTA femoral head and acetabular socket, that I will show you at least a femoral head. Now, like before or like in earlier module, that lab scale development requires that optimization of the sintering conditions, that is A, and then microstructure characterization and mechanical property like is elements of the material science. Then C is your friction and wear properties, like the way I have shown you in the last module, like in the context of the development of HDP HAP alumina composites. And this is the kind of prototype development. Then it starts with that again fused deposition method, FDM or design and manufacturing of compression mold, then ZTA uniaxial pressing conditions, those things I will explain to you slowly, and sintering and polishing conditions, about polishing of the components, optical profile and microcomputer tomography analysis of the femoral head and socket. Now, so there are some of these earlier results or earlier reports. These reports, many of them are not available in that open literature, because these are like some of the patent disclosure or invention disclosure, and these patents are filed in different countries around the world like Japan, France, USA, Germany and China. So, as you see that 5 different countries we have taken 5 different patents just to illustrate you that what is the current status of this femoral head development. Now, to mind you that these materials that they are patented, they are not mostly, they are kind of either metallic material like stainless steel or cobalt chrome alloys and so on. It has certain hardness values, elastic modulus values and strength values, or people have done some of the 90% zirconia, and one group in the USA they have developed niobium doped or niobium added alumina, niobium reinforced alumina, and they have reported the strength and elastic modulus is somewhere at 704 MPa and 55.7 MPa. One group in Germany has patented in 2005, that is aluminum zirconia, that is uniaxial and cold isostatic pressing and machining. And then another one is that in China, that is that alumina zirconia niobium and titanium 6 aluminum 4 vanadium based materials. As I said, that first step for the prototype development is the engineering design. So, therefore, I am showing you that what is that CAD design file of these materials design, and you can see that various dimensions that are mentioned, and then ultimately your aim is to translate the design concept into the prototype product by certain integrated manufacturing protocol. So, here again you have to first develop the mold where the ceramics is to be fabricated, and this mold essentially is made by that FDM technique, which is the rapid prototyping technique. You can use that ABS as the polymer, that is a very standard polymer for you, which is used in this prototype fabrication process. Now, after that you can do this prototype fabrication, then you do the green compact, and then you make the circularity and alumina as a green compact, and then you can do pre-sintering. Green compact means is a large uniaxial compaction pressure you can apply to this material femoral head, and then you can make the green compact. After that you can make the pre-sintering, and pre-sintering can be done at 1200 degree Celsius and so on. And if in the pre-sinter stage, if there is certain dimensional, if the sample goes little bit away from that original dimension, then you can do very mild machining also, because pre-sintered it is not a fully sintered, but it is good enough that you can do little bit of the machining, not that much. So, the tailoring of the intermediate machining process is very important, and then after that you can do sintering, the final sintering, and then after that final sintering, then you can do this, what is the typical wall thickness of this material femoral head. Now, one of the important thing in this entire manufacturing process is how you make the polishing of that final surface or the upper surface of this machine. This polishing is not a very simple polishing, because it is not like a flat sample polishing, but it is a rather curved sample polishing. So, therefore, you have to use certain swinging type polishing machine, polishing tool, which will alternate its position during the polishing events itself, and it can come in a reciprocatory motion, and this entire sample, entire femoral ball head also needs to be rotated during this swinging of that polishing tool itself. So, with this kind of technique, you can get that outer surface of the femoral ball head in the right polished condition. Now, this one has to measure the sphericity of this material. So, geometric circularity, that instead of sphericity, that we also call is a geometric circularity. Now, in this geometric circularity, you can find out that what is the different dimensions of these materials and how these materials is polished and smoothly polished materials. At different points you can measure this and see that what is the design file and what is the product file, Now, microstructure, one has to see that you take a one sections of this femoral ball head, and this is the scanning electron microscope image of the thermally etched sample. Thermal etching means you take the sample, you polish it, you heat treat it at a temperature which is 200 degree or 100 degree lower than the original sintering temperature for a very brief time, like 15 minutes to 30 minutes, and then this thermal etching process typically can reveal the microstructure or grain size distribution in the polished cross section. So, once you polish once you see the microstructure, once you measure the properties and so on, one of the important feature in this kind of prototype development is to quantitatively and qualitatively determine 3-dimensional macro and microstructure, and the technique that we use is that microcomputer tomography. Now, what you see in the left hand slide, it is the 3D volume rendered image of the femoral ball head. Now, 3D volume rendered image will allow you to see that how this material follows this shape and size with respect to the design file. The right hand slide image is called 2D ortho slices. 2D ortho slices will tell you these different contrast particles with different contrast. So, this is mostly the zirconia particles and rest is your alumina matrix. So, these zirconia particles, what is the size, because you know that what is the micrometer here, micrometer. Now, with the use of the several of these 2D ortho slices section and 3D cycle, you can essentially quantify that what is the zirconia particle size A, and B, how this zirconia particle size is distributed in the femoral ball head structure. Now, this is one of the assembly of that femoral ball head. This is the ZTA femoral ball head and this is the ZTA femoral socket. Now, in the as fabricated without any machining in the near net shape fabrication conditions, you can see that what is the geometric conformity of this ZTA femoral head with acetabular socket. Essentially, it shows that it is essentially good conformity. Now, just to show that how this entire assembly will look like, you can essentially make this femoral stem with the help of that ABS prototype polymer. Now, ABS polymer. then you can make this stem prototype, then you can put this stem prototype the neck into the femoral head here, and then femoral head you can see that what is the geometric conformity with respect to the acetabular socket. So, this entire THR assembly, if you see that with respect to the design file, how it is getting fitted into that actual scenario. Now, this kind of device fabrication technique, one of the things that one has to really understand that what is the typical size or what is the typical cost estimate of this femoral ball head, and then there you can see that what is the typical powders that you use, what is the cost of these powders. So, this is in the Indian scenario I am talking. Then when you make this mold, what is the cost of the mold, what is the typical labor, and what is the personnel cost, labor cost or personnel cost, and what is the machining cost and polishing cost, electricity and other things. We have done the rough estimate and we are finding the 26 millimeter femoral head cost you around 6 to 8,000 rupees, or 32 millimeter can cost you 8,000 rupees. Now, if you compare with any of these commercial available materials, this femoral ball head cost with this particular process at least 1/3 to 1/4 of the cost. Now, this is again I said that is a very rough estimate, but certainly it shows that the approach, processing approach or integrated manufacturing protocol that we have adopted, that will be scalable and that will be commercially viable, and also that is kind of a cost effective technique in the present scenario. Now, this brings us to the end of this this particular module, but before I close, that one of the things that I need to mention to you is that I need to summarize this entire course quickly, and I need to do this, I want to do this without any slides, but just to give you my own perspective. So, the first thing in any biomaterials development, you have to design that biomaterials, and design the biomaterials essentially means you have to first identify clinical need. Now, in the context of the engineering applications or orthopedic applications, first one has to interface with the orthopedic surgeon or the dentist or the prosthodontist and so on. Then you identify that what is the clinical need, then you understand that what is the clinical need, then you see that what is the existing material option. Now, once you know that what is the existing material options, you have to also know that what are the problems with the existing material options. Once you know that what is the existing problems with the material currently clinically used materials, then you have to innovate how new materials can be how to innovate new materials that you can conduct research in the laboratory and further take it further. Now, in these materials, new materials, as far as the choice of the materials is concerned, you have to see the literature and see that whether biocompatibility property of these materials are already established or not. Okay. Now, in this particular context, you have to also understand that mechanical property should not be given primary considerations, or should not be given overwhelming importance in the selection of new materials, because it is biological application. So, biocompatibility property should be given primary importance first. Now, once you do that, next thing you have to see that what is the processing approach that one has to adopt. Now, this processing approach is different for different kind of classes of materials, like metals it is different, ceramics it is different, polymers it is different. Now, processing approach you can use like conventional processing approach, and or you can use more advanced processing approach. Now, advanced processing approach you are coming you can do that rapid prototyping like 3D printing, 3D plotting and so on, or conventional processing approach can be either sintering or like compression or injection molding in case of polymer. Now, all those things essentially give you, all those things is typically used in the lab scale development of the materials. So, lab scale development essentially means that is a very simple shaped materials that you can develop without even considering that whether this approach can be ultimately adopted to develop that complicated shaped biomedical device. The way I have shown couple of examples, like one is femoral head and one is acetabular socket. What is the other considerations here? In the conventional approach, most often you cannot take the patient specific design except like compression molding mold or something. In the advanced processing approach like rapid prototyping, you can make or you can fabricate the patient specific implants. That means you can take the CT scan data from the patient and you can convert it into the rapid prototyping. We can translate the design to the product using any of the rapid prototyping techniques. So, this is the two advantages. Now, third thing is that physical property characterization. Now, physical property characterization means like in biomaterials literature often people say physical property, in the material science literature people say mechanical property. Now, physical property means you can use the hardness, you can use elastic modulus, you can use fracture toughness, that is critical stress intensity factor under mode 1 loading, and you can use several strength parameters. Now, elastic modulus by far is the most important as far the orthopedic application is concerned, because elastic modulus the mismatch in elastic modulus to a large extent can cause aseptic loosening. Okay. And therefore, as elastic modulus is one of the important factor. Strength is important because it is the measure of the load bearing capability of any implant. Now, strength can be of different this one is that flexural, one is that compression strength. In the polymer one can measure the tensile strength, or in metals also it can make tensile strength. In ceramic you cannot measure tensile strength in a conventional manner that you do in ceramics or metals or polymers, but instead what I have shown in one of the cases you can do diametral compression testing. Okay. So, I am trying to recapitulate all the things that I have taught you in several of the modules, past modules. So, diametral compression strength. Now, once you measure these kind of properties, in the ceramic special attention should be given to fracture toughness, because fracture toughness it physically tells you what is the resistance towards the crack growth by a given microstructure. So, larger the fracture toughness essentially means larger would be the resistance against the crack propagation. So, if the material A, B, C, D is there you are developing, and out of that you see that which of this material among this material A, B, C, D has the best possible combination of the bone mimicking properties in terms of elastic modulus, in terms of fracture toughness, in terms of strength. So, one material can have a very close elastic modulus to the natural bone, but that may not have the best possible combination of fracture toughness and strength. So, you have to have some balance between all these properties. You select that particular material, then you do all the detailed in vitro cytocompatibility testing. Now, in vitro cytocompatibility testing, the typical material science people, they are often satisfied with the cell viability results. But if the field, if the biomaterial science or if the biomaterial science and or biomedical engineering, the way it is today, it has developed quite far, but 10 years ago simple cell viability results would have been sufficient to publish a good paper in one of the high impact journal. But in today's world our understanding has developed and matured to extent, then one would like to see that when a biomaterial substrate, when a cell is adhering, first of all you see that what is the morphological changes of the cell, and if you remember this protein adsorption and then all those things that I have shown you, that whether there is any visible change in the cell functionality or whether there is any quantifiable measure of the cell functionality when this material when the cell is adhering and growing on the material. So, as I said that any time the cell undergoes differentiation, this is not the differentiation in a mathematical context, but this is the differentiation means differential gene expression. So, when the differential gene expression has been there and it is to be quantified. So, differentiation essentially means that is the differential gene expression. So, whether there is any change in the functionality. So, if you develop some materials which has a best possible physical properties, at the same time it can not only be used as a cell growth substrate like on which cells can comfortably grow over different time point in culture, but at the same time the same substrate can be used as the for the differentiation of let us say stem cells towards the bone cells. That is the ideal for orthopedic applications or bone replacement applications. Like those kind of substrates or those kind of materials are much more attractive compared to the conventional materials which can only support the cell growth. I hope I have made this point rather clearly. Now, once you done this cell in vitro, the next thing is that you have to see that the next thing is that in vivo. So, preclinical study is very important, and here I have emphasized in some of the lectures that animal model and defect model. Now, for the bone replacement applications, the defect model is often people use is that femoral defect model, like cylindrical defects, you drill the cylindrical hole in the femur and then you implant your material and then see that what is the bone regeneration over different time point. In another model is that what I have in some of the modules is that called segmental defect model or critical size defect model. So, all these defect models often provide complementary information on the bone regeneration capability around the material during in a particular experimental animal. So, other thing that I have emphasized in one of the earlier module and then I must repeat here that in vivo study for any of the new material that you have developed in your lab must go through a series of animal study. You have to first start with the let us say for bone it is a rabbit animal experiments. Once the rabbit animal experiments provide you satisfactory or useful results or that or establishes the tissue compatibility of the materials, then that small animal model experiments need to be validated at the larger animal model. So, first is rabbit, then you have to go to goat model or sheep model. Okay. Now, once you validate your lower animal test results in that intermediate or relatively large animal test results, then only you can go for the clinical trials in human patient subjects. Now, this clinical trials is ethically quite challenging. One has to take the approval not only from the institutional ethical committee, but also in the national regulatory committee. Once you do all those clinical trials, then only that material can finally see the light of the day, and you can transfer the technology to a biomedical company, and you can take that before that you can take the national regulatory committee approval like FDA in United States or similar regulatory approval in other countries, and then a company can produce the materials in multiple numbers and sell it to the hospitals and so on. So, I think with this course I have covered a broad spectrum of knowledge with you, so that you are in a position to design your experiments, to understand and analyze your results in the light of the biocompatibility evaluation, as well as design new materials and lastly new implants and devices, so that this research becomes very useful to the society as well as a larger cross section of the patients.